Thank you very much for inviting me. My friends who are at your meeting right now gave me some helpful hints. Vlad told me I had to speak in Russian, which I won't do. And Elena told me that it was a very broad crowd with a lot of different scientific interests. That always makes it difficult because however I structure the talk, half the people are going to be bored. So I took the Army approach to this, and I'm going to split it in half so that everybody is bored for part of it, but everybody understands the other half. So what I want to talk to you about is nucleoside-modified mRNA-LNP Therapeutics. My disclosures, it's a bunch of patents and a few companies. Let me start with the background of mRNA. So mRNA was first identified in 1961. The first time it was injected into an animal with the idea of using it as a therapeutic protein delivery system was in 1990. There was a single report after that in 92, and the rest of the studies looked at it as a vaccine, but a vaccine where they took dendritic cells out of animals or out of people, pulsed them with mRNA, and then gave them back. And there was a reason why that was done. And that's because of this. And this is not the right, sorry, slides are a little mixed up. Let me, so the SARS-CoV-2 vaccine, let me give you some background first. The virus looks like this, and it's composed of a membrane derived from the cell that has spike proteins on the surface. And we've all heard about the spike protein. It's the protein that mediates infection of respiratory epithelial cells through the ACE2 receptor. And it's also the immunogen that all vaccines are using. But the spike protein is interesting. It exists in two conformations. In one, the receptor binding domain, the part of spike that binds to the cells, is hidden. And in the other, the receptor binding domain is exposed. It can only infect cells when it's exposed, and it can only be neutralized well when it's exposed. There are many vaccine platforms that have been developed very quickly over the past year. And over 111 clinical trial developments are going on. But looking at vaccines more broadly, the first vaccine that we identified was in 1798. And this was a vaccine developed by Edward Jenner. And what he did is he noticed that milkmaids during the smallpox epidemic who were working on cows that had pox lesions that looked like smallpox weren't getting infected. They were protected. So his idea was that he took the insides of the cowpox lesions and used those to immunize people, and they were protected from smallpox. That's our first identified vaccine. But as you can see, many other vaccines using a variety of platforms have been developed. Inactivated pathogens, which are characteristic of influenza, proteins, the tetanus toxoid is what most people get every 5 years, and newer platforms with viral vectors, DNA, and RNA. So what I want to talk to you today about is the RNA vaccine platform. And this is an overview of the platform itself. And this is used by the WHO in describing vaccines. So mRNAs have been in clinical trials. So they've been given to people for the past 15 years. It's not a brand new technology that was invented in 10 months. It's been used clinically for a variety of diseases. Currently, there are 6 COVID-19 vaccines in clinical development, and the first 2 RNA vaccines have been approved for use around the world. People like the mRNA platform because it's very immunogenic. That means it makes very strong CD4 and CD8 T cell responses and neutralizing responses. It's very potent. It's also very safe. The RNA does not go into the nucleus. It does not affect DNA. It has a very favorable large-scale use. We've given it to nearly 1 billion people with minimal adverse events. It's given intramuscularly, usually as 2 or 3 doses. The early concerns were that it was difficult to store. It required minus 80 degrees centigrade freezing. That has largely been solved. The other thing that people like about it is it allows very fast development. So you only need the sequence. You don't need to isolate the virus. You don't need to grow it. You just need the sequence of the immunogen of interest. It can be rapidly scaled up, producing, I think we're at 50 million doses a week right now. The way the vaccine works is that you start with a DNA template. The DNA template encodes a promoter for a phage RNA polymerase, the coding sequence of the immunogen of interest, and other RNA support structures, untranslated regions, poly A tail. You then put it in an enzymatic reaction with the phage RNA polymerase. The polymerase reads the DNA and makes an mRNA copy of it. You take that mRNA, you purify away contaminants, you load it into a lipid nanoparticle. The lipid nanoparticle protects the RNA from RNase degradation. When the cell sees those particles, they're endocytosed, they're eaten by the cell. The RNA escapes, it makes it to a ribosome, which produces protein. The protein is either put on the surface of the cell or processed for MHC class 1 and class 2 presentation to T cells. There are many optimizations that make the vaccine and the RNA work better. So you have to optimize all of the components of the mRNA, the cap, the untranslated regions, the coding sequence, the poly A tail. You then have to optimize the lipid nanoparticles in order to have high encapsulation and efficient delivery and adjuvanticity. So I'm now going to move on to the basic science of why the mRNA vaccine works so well. And I apologize to the physicists in the audience, but I'm sure the internal medicine and immunologists and virologists will be happy. So back into why RNA wasn't used any further was because of this. There are 17 innate immune sensors that recognize RNA as foreign. They produce inflammatory cytokines, type 1 interferons, they induce apoptosis, they directly block translation, and they induce cell death. So the work that Katie Carrico and I did, and this is starting around 1998, was we wanted to understand what made RNA immunogenic. And we were the first to identify that RNA was highly inflammatory when given to cells. And that was completely unexpected because our cells are full of RNA. And there was no reason to believe it would be seen as foreign. So this was one of our leading studies. And what we did is we took RNA from a variety of sources, mammalian and bacterial cells, and then we split them up into subfractions. And the question was, if RNA was simply immunogenic, then all RNAs would have equal immunogenicity. But that's not what we saw. Bacterial RNA was highly immunogenic, and transfer RNAs from mammals were not immunogenic. That led us to the hypothesis that nucleoside modification could be involved. And that's because bacterial RNA has fewer modifications, while transfer RNAs have up to 25% of their nucleosides modified. There's over 100 different types of modifications. They include methylations of sugars and bases. They're at conserved positions, and it's recently been shown that M6A is reversible. So we made nucleoside-modified mRNA. In that enzymatic reaction, instead of putting in UTP, we put in pseudouridine triphosphate. So every U has pseudouridine, or every C is methylated. And what we found is that that reduced the inflammatory activity of the RNA. We reduced how much TNF and IL-12 was made by cells. But the striking thing, and also unexpected, was that the modified RNA was translated better. In this case, it was about 10-fold better than unmodified RNA, which was completely unexpected. We also had to develop a purification because the enzymatic reaction made double-stranded RNA contaminants, which inhibited translation and induced inflammation. This is the combination of nucleoside modification with purification of double-stranded RNA. And what you see is that there's a 4-log increase in the amount of protein made from the same amount of RNA, an enormous increase. Now, we're talking now 2003, 2004, and people asked us, well, who cares? Why are you working with RNA? It's difficult to work with, it's unstable, it degrades very quickly. People tried it before and it doesn't work. And our response was, protein therapeutics are the most advancing field in pharmaceutical science. Largest growth is in protein therapeutics, many of them monoclonal antibodies. This is what a GMP site looks like for the production of human monoclonal antibody therapy. You have 50,000-liter drums full of CHO cells or other cells that secrete the protein you want. You then have to invent a way of purifying that protein away from the cell culture media, the cells, and any viruses that might be mixed in. With mRNA made in a single reaction vessel. Moderna and Pfizer use 100-liter bioreactors. The procedure for purification is one vessel. Those are identical no matter what the RNA encodes, what the coding sequence is, the protein that it makes. So it's a much simpler production. It's much easier to scale up large scale. Now, the other issue is you have to deliver the RNA. Just about every fluid and every cell in our body has RNases that rapidly degrade RNA. So we studied over 40 different formulations and identified lipid nanoparticles as the best. Lipid nanoparticles contain 4 lipids that self-assemble with mRNA. They bind ApoE, and ApoE is a protein that targets liver cells for delivery of lipid material. They're very safe and very effective. We put mRNA into a lipid nanoparticle and injected them into mice. And what was striking is that peripheral injections, intradermal, intramuscular, subcutaneous, had long-term translation of the mRNA, up to 14 days. That made us think this could be a good vaccine. A good vaccine requires long-term production of the immunogen to load the lymph nodes, the germinal centers, and make good B-cell responses. So we started with influenza, and this is a review of mice immunized with influenza vaccines. And what you can see is that with just about all of them, 2 immunizations with live virus, inactivated virus, purified protein, gives you an HAI titer, which is a measure of neutralization of around 1 to 100. Protection in humans is seen at a titer of 1 to 40. So that's why we think these vaccines are good. We then studied the mRNA version of the vaccine, and we're comparing it to inactivated virus and live virus. The mRNA vaccine gave titers of 1 to 2,500, so 25 times better than other vaccine platforms. In vaccinology, that's a striking increase. Now, we needed to understand what the mechanism of this was. This is a cartoon of how antibody responses are made. So a follicular B cell in the lymph node encounters a virus, antigens, innate immune adjuvants, binds them by the antibody on the surface of the cell, and gets help, cytokines, signaling molecules from CD4 helper cells. They then become germinal center B cells. These are the cells that rapidly proliferate and affinity mature. So they increase the affinity, the binding capacity of the antibody. They then turn into plasma cells and memory B cells. Plasma cells produce the antibody long-term. Memory B cells are there for the next time you see the immunogen. We used an antigen-specific assay. So these are B cells that bound hemagglutinin that had a fluorescent tag. We're only measuring antigen-specific, HA-specific B cells. But what you can see is that compared to an inactivated virus, all of these populations are about 50 times higher, 50 times more germinal center memory and plasma cells than an activated virus. That explains the increase in titer. What was striking, though, is we left mice for 13 months after a single immunization and measured the plasma cells in the bone marrow. This is where B cells go to produce antibody long-term. And after a year, we found high levels of plasma cells making antibody in the bone marrow. So the question then came, well, how is this vaccine working? And I started off by telling you that nucleoside-modified mRNA has no adjuvant activity. It doesn't induce inflammation. It doesn't induce inflammatory cytokines. We know that vaccines need adjuvant to work, and they're typically described as Th1 or Th2 inducing. But we were focused on a different kind of CD4 helper cell, known as a T follicular helper cell. And what these cells do is they form germinal centers. And in the germinal center, the germinal B cells rapidly proliferate, affinity mature, class switch, and form memory and plasma cells. If you don't have T follicular helper cells, you get a lousy antibody response. We measured the T follicular helper cells in immunized mice. This is now the total number, and you see about a 10-fold increase in mouse spleen and lymph nodes. We also looked in macaques because with any vaccine, you want to be sure it works in larger animals. And we also had an assay that measured antigen specific. So this is measuring T follicular helper cells that recognize, that bind the envelope antigen. We compared it to another adjuvant that was supposedly good for TFHs, but you can see the mRNA-LNPs induced a huge increase in T follicular helper cells. So that gives us the mechanism of why the vaccine works. The first is that the RNA-LNPs produce protein for an extended period of time, which continuously loads germinal centers and makes a better response. And second, the LNP adjuvant induces T follicular helper cells, which are the best cells for driving antibody responses. But we also found something else out about RNA vaccines, And in this figure, we wanted to understand what the vaccine was making antibodies against. So the head domain of hemagglutinin is the part of the molecule that binds respiratory epithelial cells and infects cells. It's also the part of the HA that rapidly mutates and makes us invent a new vaccine every year. The stalk domain catalyzes viral envelope fusion. It's more conserved. It's also subdominant, which means you don't make antibodies against it very easily. With most vaccines that are used, you get head responses, you don't get stalk responses. So we studied this, and what we did is, in this well, we put hemagglutinin where both the head and the stalk were from H1, which is the same strain as the vaccine. In this well, we put an H6 head with an H1 stalk. The H6 head is not recognized by H1 HA immunized animals. So we're only measuring stalk binding here. And what we saw was really enormous responses to the stalk with mRNA-LNPs. We wanted to know, could that lead to protection from infection from distant viruses? And as we all know, influenza vaccines currently used on average are 50% effective. And a lot of that is because you choose the virus 9 months before you make the vaccine. And if the virus changes, if it picks up a mutation here or there, the vaccine stops working. That's called drift. So we tested drift by immunizing with the 2009 H1 virus and infecting with a 1934 H1 virus. There's no head cross-reactivity. The animals were completely protected. We made it harder. We immunized with an H1 and challenged with an H5, so a completely different subtype of virus. Current vaccines give no protection against H5. The animals, again, lost no weight, were completely protected. So what this means is that the influenza mRNA-LNP vaccine is incredibly potent, much more potent than other vaccines currently in development. A single immunization protects. The mechanisms are potent TFH induction and extended production of immunogen. This is now being studied as a universal influenza vaccine because it protects against drifted viruses and it protects against new pandemic strains. So I just wanted to end by talking about some new physiology. And this is actually physiology therapeutics that were developed by Vlad Muzikantov, who is in the audience right now. The way the LNPs are made is the lipids are in ethanol, the RNA is in acidic water, you mix them together and they self-assemble into a lipid nanoparticle. These LNPs bind ApoE and target the liver. We've also shown they target dendritic cells, which is why they're so good at being a vaccine. But the question is, what if you want to target a different cell? What if you wanted to deliver RNA to a different cell type or a different tissue? You have to develop that. So what Vlad's group has done over the years is they've developed a way to link targeting antibodies, targeting ligands, receptor ligands to a PEG lipid that can be inserted into the lipid nanoparticle. So now the lipid nanoparticle has an antibody coating that can target anything that the antibody binds. They found when they did this, the LNPs didn't change morphology. They were a little bit bigger. This is about the size of an antibody coating. The surface charge didn't change. But most importantly, they were functional. So this is binding. This is PCAM-targeted LNPs binding to PCAM-expressing cells, not binding to non-expressing cells. This is the luciferase mRNA in the lipid nanoparticle. After it binds to PCAM-expressing cells, the RNA is delivered and it's translated. These are UVECs that express PCAM, and you only see mRNA expression making green fluorescent protein when they're targeted. When the LNPs were targeted to PCAM and injected into an animal, the lungs lit up. The LNPs went to the lungs. They targeted the lungs because they have the highest density of endothelial cells expressing PCAM. So we were able to develop specific targeting of LNPs to things other than the liver. Now, my lab has an interest in T cells because we study HIV. We did the same thing. We attached CD4, anti-CD4 antibodies to the LNPs. They bound T cells very well. We also showed that the T cells express the mRNA. And that's difficult because T cells have no endocytic activity. The hypothesis was if we target the LNPs to an antigen that endocytoses, that goes into the cell after binding, it would deliver the RNA. We then moved to a system where if we delivered a protein, the protein acted on the DNA and made the expression of a fluorescent protein. Here, the targeted LNPs are expressing the protein in around half of the T cells. When we injected them into the animal, the spleen lit up. That's where the T cells reside. But importantly, we imaged the mice again. So these mice have had all of the previous organs removed, and we imaged them again. And what we saw was that lymph nodes were now showing up. So what this means is that the LNPs are injected into the bloodstream. They escape the bloodstream, go into the tissues, go into the lymphatic drainage and drain into lymph nodes, and are still able to target and deliver RNA to T cells in lymph nodes. We've developed a new type of therapy using these LNPs, and I'll end my talk with this. So Penn developed CAR T cell therapy clinically. We've had a couple that have been FDA approved. The way this is done is you take a patient with usually a lymphoma or leukemia, you leukapherese their cells. That means you take many cells out of their blood. You stimulate the T cells for 10 plus days, infect them with a lentivirus that delivers a CAR molecule. The CAR molecule targets a specific antigen expressed by leukemia cells. Typically it's CD19. And then it has a tail in the cell that signals the cell to kill. So it makes artificial killer cells. It's very effective for treating certain leukemias and lymphomas. But the procedure cost $500,000 per dose. We wanted to see, could we make CAR T cells in vivo? So what we did is we took an mRNA-LNP, we put a CAR molecule in the RNA, and put a T cell targeting antibody on the surface. And the idea would be that the cell will take up the LNP, the RNA is released, translated, and puts the CAR molecule on the surface that can then attack cells, in this case fibroblasts, that expressed what the CAR recognizes. This procedure efficiently modifies T cells, so 80% of the CD4s and CD8s are modified with T cell targeting. And we can efficiently deliver CARs in vivo. 20% of the T cells after a single injection have the FAP CAR on their surface. We then use these FAP CAR T cell therapies in a model of cardiac fibrosis. So the mice are made hypertensive, and hypertension leads to cardiac fibrosis. And you can see in the yellow bar, fibrosis leads to systolic and diastolic dysfunction. The heart doesn't work as well. The ejection fraction, the amount of blood the heart ejects, is decreased. We treated them once in the red bar with CD5 T cells expressing FAP CAR that made CAR Ts in vivo and attacked the fibrosis. And we were able to get rid of the fibrosis and return cardiac function to normal. So this means a single injection of mRNA-LNPs were able to treat a disease that required a $500,000 therapy and weeks of development prior. And we see this as a future. The development and the delivery of mRNA-LNPs in vivo opens up brand new therapies. It can be used for gene correction. We can deliver LNPs that target bone marrow stem cells to treat sickle cell anemia. Intellia has already treated amyloidosis in the liver with LNPs. It's a huge potential for the future. In addition, we're working on many different vaccines. This is just a subset of the diseases, but of interest includes things like malaria, TB, universal influenza, and pan-coronavirus vaccines, as well as many other therapies that are currently in development. So I need to thank all of the people that were involved in these studies. Norby did many of the vaccine studies. Hamada did a lot of the targeting in Vlad's lab with Vladimir Shuvov as well. John Epstein's lab did the cardiac fibrosis work and all of our funding. Thank you.